Hey everybody, so today I want to give you some quick information on how viruses attack. The reason I want to talk to you about this is because it's common to use the term life cycle with a virus. And remember, viruses don't have the characteristics of living things, so they can't really be considered living, and that's a really confusing thing for students because if you look at viruses, and we know that viruses are active inside of us, it makes us think that they're doing living functions. They look like they have behaviors, but they're not living. So let's look at viruses in a little bit more detail, and I want to talk to you about how they attack. Keep in mind kind of the analogy in your head of a robot, right? So a robot might appear to be living, but a robot can only do what it's programmed to do. So it might look like it's carrying on life functions, but it's not made of cells, it doesn't have homeostasis, it can't grow, it can't reproduce. All those things in that characteristics of living things lesson that you guys had, right? So think of a virus like a robot. And instead of a program, what tells the virus what to do is the DNA inside of the virus. And I know you know about that because we talked about viral structure in one of the lessons. So let's jump in then to how does a virus actually attack a cell, okay? And we're going to start right here. This is why I like to call this the viral cycle instead of the viral life cycle. This is the basic, whoops, and there's a misspelling here. This is the lytic cycle, L-Y-T-I-C cycle, the lytic cycle. Because what happens here is, step one, the virus attaches to the cell and injects its DNA, and that's what you can see right here is the DNA from the virus being injected. Once the virus attacks, its DNA enters and gets spliced into the host cell chromosome like that. Okay, so you can see, it's kind of hard to tell in this picture, but the viral DNA is a piece of red DNA that's kind of right there in that plasmid that you see. And it's attached to the host chromosome. Now, in the third step, replication can happen. So the viral DNA can get copied along with the host cell's DNA. Okay, so that's step 3, replication. Step 4, translation can happen. So viral proteins start to get made. So the little red things are the viral DNA here. These little blue things are the protein coats of the new viruses. Step 5 is assembly. So now we start to stick the DNA into the virus, the protein coats of the new baby viruses that we've just made. And step 6 is release. So what happens is the cell gets so full of viruses, it gets pumped up until it bursts open, and that's called lysis when that happens. And that's why this is the lytic cycle for viruses. So essentially in this cycle, viruses attack, they reprogram the host cell, they force the host cell to make new copies of DNA and new copies of viruses, and then once the cell has produced so many viruses it can't hold them anymore, it pops open and out go all of the viruses. So that's a lytic infection. And examples of viruses that do this are rhinovirus, which causes the common cold, and influenza. Okay, so these are viruses that you catch, you get them, you're sick with them, and then once you're done being sick with them, you're done being sick. The virus goes away. So your immune system can fight these. Now remember, to cure one of these, if you want to cure it and you don't want to wait for your immune system, you're going to have to take an antiviral. So if you have the flu, maybe your doctor would give you Tamiflu or something like that. All right, moving on, let's look at a more complicated cycle. So we just saw the lytic cycle, which is up here at the top, right? Here's the lytic cycle. But that's not the only viral cycle that there is. Some viruses are really sneaky. Examples of viruses that are like this are things like herpes and HIV. The chickenpox virus can do this as well, which is a form of the herpes simplex virus. Human papillomavirus, which is the virus that causes warts. All of those viruses can get into your system and stay there. Once you're infected with them, you have that virus for the rest of your life. So we're still starting here at number 1, but now when the DNA gets inserted into the host chromosome here at step 2, it doesn't become active right away. So it just sits there. And the thing is, if this were a body cell, the person's immune system can't really detect DNA inside of a cell as being a bad thing, right? DNA is supposed to be inside of cells. So the host immune system can't tell the difference between viral DNA and the DNA that's supposed to be there for the host chromosome. So what happens is the virus can actually hide out this way, undetected by the immune system. So step 3, when the cell gets ready to divide, it's going to copy the viral DNA and its own DNA. See, it's in there, right? Here's the little plasmid with the little red viral DNA. So as the cell divides, and it's kind of here in step 4, both of these are step 4, you'll see the replicated DNA now ending up in 2 cells. When those cells grow up and replicate their DNA and divide, then you get 4. When those cells grow up and replicate their DNA and divide, then you're going to get 8, and then it gets to be 16, and then 32 and 64 cells, and eventually you've got this big patch of cells that has this virus DNA inside of them. And it's undetectable, it's hiding there, until there's a trigger. Now, triggers can be anything that weakens the immune system. So anything that causes the immune system's back to be turned for just a little bit is going to cause this lysogenic virus in some of the cells. Say like this cell right here, it's going to become active. So the virus would then pop out and go here and start replicating, okay, and then start translating and start assembling and then release. But not all of the cells get triggered at the same time. Let's talk about triggers. Cold sores are a great example of this type of virus. This is a herpes simplex virus that's on your lip, right? And if you get cold sores, you have this virus in all of these cells. And they're called cold sores because you get them when you have a cold, or sometimes older people in the South call them fever blisters. And again, it's because your immune system is fighting another infection someplace else, a fever someplace else in your body. You have a cold right here in your nose, and so your immune system's fighting that, which gives the virus on your lip the perfect opportunity to pop out and become active. Other things that could trigger these are stress, lack of sleep, lack of exercise, poor nutrition. All of those things are going to weaken your immune system and cause one of these viruses to pop out. So if you're somebody who has cold sores, you know that you probably have some triggers where that comes out. Maybe if you get stressed a lot around exams and you get a cold sore, that's probably why your immune system is a little weak and the virus pops out then. But again, because not all of the viruses trigger at once, you still have some cells, like right here, like this one, that has a virus inside, but the virus is still dormant. So, like I said, once you get these infections, you'll have them for the rest of your life. There's no way to get rid of a lysogenic cycle virus, okay? So if you got it, you're always going to have it. And that's how those viruses tend to operate. So that's how viruses actually run their program, and that's what they're programmed to do: get inside of cells and reproduce. Whether it's by the lytic or the lysogenic cycle, that's what they do. They run that program and they just reproduce and reproduce. So they attack cells and get the cells to reproduce for them. Notice, though, they don't ever grow. They're manufactured to be the same size that they'll always be. They don't have homeostasis. They can't reproduce without getting into a cell and using somebody else's reproductive machinery. So again, it's kind of like a robot. They're running a program, but they themselves are not capable of living function. So remember, viruses are not alive. Bacteria, that's another story, and we'll get into that tomorrow.